Oh yeah, it says pause stop recording, so I think it's recording.
Where do you see that?
Like the top.
Actually, it's true. It's the timing already.
That says you are screen sharing.
Like right here it says pause to stop.
Oh, thank you. Okay. All right. I think we'll get started. You can see our screen, our slides as well.
Yes.
Great. All right. Okay. Welcome and thanks for tuning in. And my name is Matt Koski. I'm an associate professor at Clemson University in South Carolina in the Biological Sciences Department. And we have been running a community science project called the Clemson Flower Color Project for the past 2-ish years now. And my lab traditionally doesn't do community science, so this has been a work in progress, and I think we're getting there. And I am going to go to the next slide, and we're all going to introduce ourselves. There's been several members of the team over the years, but right now you're looking at the current group that's working on this project. I'm already kind of introduced myself, but I'm Matt Koski, Associate Professor in Biological Sciences at Clemson.
Yeah, so I'm Mercedes. I'm a postdoc at Clemson University with Matt Koski, and I joined the project this year.
I'm Grace Everson. I'm a biological sciences undergraduate student, and I also joined the project this year. My name's Zarelli. I am an undergraduate biology major at Clemson, and I also just joined the project this year.
And I guess, yeah, if you two, the two that are here, want to introduce yourselves as well, that'd be great.
Go ahead, Richard. I'm just a hanger on.
Yeah, I'm Richard Thunberg. As Sally said, master naturalist in Virginia and board member and newsletter editor for the Piedmont chapter of the Virginia Native Plant Society. And you contacted me 2 years ago because you found my pictures of Phacelia dubia on iNaturalist. So I said, okay, I'm always out there taking pictures. So I signed on. And,
Yeah.
I'm glad you made it easier to put up the pictures this year than last year.
I think it has helped and increased participation a lot. So.
And I'm Sally Anderson, and I sit on the Piedmont chapter board with Richard, and I'm the second vice president for the state VNPS and a past president and a master naturalist, and I have nature ADD. That's
So
it.
sent you a color checker card yet?
No,
Yeah.
I don't take anywhere near as many pictures as Richard does, but if I get interested today, I might do it. So.
Okay. Well, thanks for your intros. We're gonna now just kind of get a bit into the background of the project and walk through some of the questions that we're using the data that you all submit to answer. All right.
So our lab started looking at flower color variation within species. As you can see in these photos, flower color can vary a lot within a single species, but we really want to know, why does this change in pigment occur? So there's different factors that can influence flower color. One, for example, is our biotic factors. So that would be pollination, pollinator selection. So what flowers are selected by the pollinators, and then we also have abiotic factors, which can also affect the flower color. And so, like, for example, we have temperature, water availability, and solar radiation. All of these can affect flower color. Like, for example, dark flowers can absorb more heat, which can lead to more reproduction, biological success, and then more dark flowers following that. So when we have pollinator selection, this gets informed by the pollinator visual system. So we see on the blue, green, spectrum, and pollinators also see the ultraviolet spectrum. However, we looked at the pollinators that we looked at, and they actually don't use the ultraviolet spectrum for selection. That's right.
No, so we picked flowers that don't reflect in the UV. Yep.
So our flowers don't reflect in the UV, so that is not a part of it, but we do for what we're looking at, look at the blue and green spectrum. And so this affects pollination because the pollinators can see the flower color differently than we do, and their preferences for flower color will depend on their perception of the colors. So we are also looking at the contribution of how background color can influence the flower color selection by pollinators. So if you look, which colors do you guys think are more conspicuous in each background? Like, for example, the light background, which color is more conspicuous?
So if you were to squint your eyes and look at this light green background, which one do you think is popping out more?
They actually sort of pop out equally to me, but that's not true of the other one.
So most pollinating insects have really coarse visual systems in terms of like visual acuity. So if you were to hold out your thumb in front of your face and look at your thumbnail, they see the world roughly pixelated in the size of your thumbnail.
Oh, wow.
Yeah, so they're not seeing tons and tons of detail. So I guess, okay, so looking at the bottom one, which one pops up,
Oh, definitely the pink.
right?
Yeah, yeah.
How about
So,
you, Richard?
so what we're noticing is that the darker colors tend to pop out a little bit more on the light background, and the lighter colors pop out more on the darker
Yeah, I
background.
can see
So like as you can see, the darker colors are more visible against the light background.
I get it.
And then same thing for the dark background, the light flowers are more visible against that. So we would expect that this is how the bees are seeing it as well, that the dark flowers are more conspicuous to them on the light background. And same thing.
Yeah, so over many generations, for example, if you have a population against this dark background and there's flowers that vary in color, if pollinators are continually choosing the lighter colors here, they should impose selection to lead to more lightly colored populations. And the opposite may happen in the lighter background.
So these are our current project motivations. Our two main goals are to assess whether flower color can evolve to contrast with the local background, and also determine if there are broad geographic patterns in flower color diversity. And our question is to see if flower color can evolve to increase its flower color can evolve to increase conspicuousness to pollinators against the local backgrounds. So currently, this is our progress summary in terms of our photos and our populations. So as you can see, we have a good amount for these 4 species of photos of Histonia caerulea, Histonia priscilla, Hydrophyllum virginium, and Phacelia dubia, and a very large amount of populations for these across our photos. And then the 4 other species that we look at are Vistigia virginia,
Pistostigia, probably.
Pistostigia virginia, Viola pedata, Iris verna, and we have a bit less photos of these species and less populations that we're looking at. So we're looking to increase these as well as the other species that we're looking at just to get more data for everything.
Yeah, and so we started out with maybe 2 or 3 focal species in the first year, and we've added several species moving forward. So some of these, like Pistostigia, Lobelia syphilitica, were added, and Viola pedata were added later. And so that's maybe some of the reason we have fewer photos and populations for those ones.
So right now you can see sort of our distribution of our populations. Currently, you can see we have our one map that shows over the years where populations are concentrated. So a lot on the southeast and then up in Wisconsin mostly. And then on the second map, you can see the different species. So like you can see we have a lot of Phacelia dubia and large concentration of the Hydrophilum up in Wisconsin and a large one of the Histonium. But we're just looking to increase the amount of population data across all over the southeast and see this big area that's all blank and missing data for right now. So yeah.
Yeah, so if you all have friends or know people in other native plant societies or other naturalists that are in some of these other regions, a lot of the species that we're working on are pretty widely distributed throughout the East. And so we would love to kind of fill in geographic gaps by recruiting more folks to this project.
Okay, so what do we do with your photos? So once we upload them from iNaturalist, we upload them into Photoshop, and I'm gonna take this from you. And we use the color card to set the white standard. And what this does is it allows us to extract an accurate color no matter what lighting your photo has. From there, we choose 2 flowers, preferably from different plants, and from each flower, we extract the color from 1 petal, and we use the tip just to get an accurate color reading. And we extract the red, blue, green color from this. Once we do that, we divide the photo into 4 quadrants, and we extract the color from a random location in the background. And we try to avoid things like if your thumb accidentally gets in the picture or if there's trash, because obviously those colors wouldn't occur in nature. So we just try to avoid that. Now, what do we do with the red, blue, green color that we extract? We try to find the green chroma. Now, what exactly is the green chroma? The green chroma is the intensity of green reflection from an object. And to do this, we take the overall green and we divide it by the red, blue, green color. And this is important because pollinators use green photoreceptors to distinguish flowers from the background. But in addition to the green chroma, we try to, oh, sorry, I got ahead of myself. So this is an example of 2 backgrounds with different amounts of green chroma. In this one, it would have a large amount of green chroma, and then this background would have less green chroma. Okay, so we also use the red, blue, green data to find the blue chroma. And the blue chroma is the intensity of blue reflection from an object. And to find this, we take the overall blue and we divide it by the blue, green, and red. And this is important because it tells us how much pigment is in a flower. Okay, so now there are some photos that are a little bit more difficult for us to extract the color from. These could be photos with shadows because it just causes an inaccurate color, blurry backgrounds. A slightly covered color card would make it hard for us to set a white standard. And then any photos containing flash can change the reflectance of the flowers. But this doesn't mean that we will not use photos that contain any of this criteria. It just takes us a little bit longer to extract the color, but we still appreciate any photos that you guys send to us.
And just a quick note, this top photo of Iris with super mottled light, I took. And so you can see like the white standard is kind of in this bright light, and then some of the petals are in lower light. So that changes some of the measurements and that contributes to error in the data set. But I'm just mentioning like, I took these and I'm helping to manage the project. Sometimes you can't avoid some of these situations, but if I were to do this again, go back to the site, I would probably take the color checker card, put it in bright light, and take the photo of these 2 flowers, and then take the color checker card and put it in the shadow and capture these flowers. So anyway, just a note that we still can use these. Yeah. They're just a little off.
And so does an overcast day matter? Is that better or worse, or?
That's fine.
That's fine.
And it's just more so that if there's like differences in the light across the photo,
Yeah.
Oh, okay. All right.
all in one, like bright light or one overcast, that's fine.
Yeah.
And actually, what the standardizing is helping to deal with is what you're suggesting. So, like, some people are going to be out on cloudy days. That's fine because we're using that white standard on that day. And then some
Yeah.
people will be out on really bright days, and having the color checker card in the photo helps us deal with that variance.
Okay. So now we're going to move into the preliminary results. We're going to be showing you guys the results for 3 species, Hostonia caerulea, Hydrophyllum virginianum, and Phacelia dubia. And we chose these 3 specifically because these are the species where we have the most data. And we're going to be looking at the relationships between flower chroma and background chroma values. We're also going to be looking at spatial patterns, so longitude and latitude versus the blue and the green chroma. Okay, so first we're going to look at Hostonia caerulea, and we're going to look at the green chroma in the flower versus the green chroma in the background. So the graphs show it's per population, so every dot is a population of Hostonia caerulea. And this graph showed a positive correlation between the flower background and the green chroma. So what does this mean specifically? So a greater green chroma and flower petals means that the flowers are going to be lighter in color. The reason why this arrow is blue rather than green is because the green chroma doesn't necessarily make a flower petal green, it just makes them lighter in color. So for example, down here would be a flower with very light green chroma, and you can see an actual example in a flower, and this flower would have greater green chroma. So what does this graph specifically say? It says that populations with a higher background green chroma have lighter flowers. So you can see here where the arrow is darkest, if you go up here, the flowers are going to be lighter in color. Okay, so now this is also for Hostonia caerulea, but this one shows the flower blue chroma versus the background green chroma. And again, this is per population of Hostonia caerulea. So this graph shows a negative correlation between the flower blue chroma and the background green chroma. So flowers, when flowers have less blue chroma, they tend to be lighter in color. So this graph is telling us that populations with a higher background green chroma tend to have higher background tend to have lighter flowers. Oh yes.
Yes.
That is correct. Sorry, I confused myself for a second.
Yeah, so this plot is showing basically similar results as the other one because that as much like more green chroma in the flower means that they are lighter, but more blue chroma in the flower in this case means that they are darker.
Okay. Okay, so now we're moving on to longitude and latitude versus the flower blue chroma. So this is for Phacelia dubia. And again, this is per population. And so when we're looking at the blue chroma versus the longitude, we can see a positive correlation. So populations that are further east tend to have darker flowers, and populations further west tend to have lighter flowers. Now, when we look at the blue chroma versus the latitude, populations further north tend to have darker flowers as opposed to populations further south, which have lighter flowers. Now, looking at Hydrophyllum virginianum, we're also looking at the blue chroma versus the longitude and latitude. This one is different from Phacelia dubia because flowers located in the east tend to have lighter flowers, and flowers located in the west tend to be lighter in color. Now, the blue chroma versus the latitude shows us that flowers further north have darker petals, and flowers further south have lighter petals. Okay, now just to recap, Houstonia ceruleus showed that populations are lighter, meaning they contain less blue chroma when they have greener backgrounds. For Phacelia dubia, populations further northeast are darker, and then for Hydrophyllum virginianum, populations further northwest are darker. So what does this show us? So it shows us that flower colors shift with background conditions and locations. And we also saw that for Phacelia dubia and Hydrophyllum virginianum, flowers that were located north had a darker color. This can probably be due to the fact that darker flowers are able to absorb more light, which would help them thrive in the colder environments that can be up north. These trends support the idea that flower color evolves to stay visible to pollinators and matches local conditions. Now, in the broader spectrum, what could this help us with in the future? So as climate change alters vegetation, temperature, and pollinator activity, understanding these patterns can help us predict how plant-pollinator interactions may shift in the future.
So these are some kind of, like I said, we were talking about very preliminary results. And so we're hoping to add more photos to this and analyze the entire data set across all the species. We were just giving a smattering of some interesting patterns that we were starting to see. We have a project website. So for example, Sally, if you wanted to have us send out a color checker card if you're out taking some photos of those species that we listed, take a look here and it has our email and information on how to get a color checker card to participate. And it's all linked through iNaturalist. So we, like Richard said, we used to have a pretty clunky submission system that we've changed and made it a lot easier for folks to participate. And then the students, the youth, have started an Instagram account for the project. So hopefully we'll keep that updated if you want to follow along. So here's a list of all of the people across several states that have contributed photos to the project. So huge thanks to all of the people out there taking photos and submitting them for the project. Here's a list of past students that have been part of the project at Clemson. And a big thanks to Clemson's Creative Inquiry Program, which is an undergraduate research-based credit program, and then the National Science Foundation that funds this project. And yeah, I think now we are open to any questions you might have or thoughts about the preliminary data or anything at this point.
Well, I have in the back of my mind somebody doing research on a plant that it's been so many years ago, I can't even quite think what it was, but they were doing genetic research and they found different populations of this plant in certain parts of the East based on genetics. Do you think this is related to that, or do you think they're like kind of separate populations that have evolved in their own way, or do you think that there's interbreeding or?
Yeah, that's a great question. So plants can upregulate the compounds that make the flowers more purple or in certain conditions and like cold temperatures or stressful drought conditions. And so that would be like some of the differences we are seeing among populations could be due to what's called plasticity, like similar genotype expressing different phenotype or color in different environments. And then some of the differences between populations could be due to like fixed genetic differences, like they're not sharing genes between populations. And with this data set, which is all very correlative, like we have, location and color. We don't have information about the genetics. We won't be able to pick that apart. However, the lab part of the National Science Foundation project is focusing on Phacelia dubia as a model system. And we're getting a lot more information on that in terms of understanding whether color is genetically determined, how populations differ from one another genetically, and if that shapes color differences. And what we can say in that species is that there is genetic variation for color. Like if measure the color in the field, and then we collect the seeds and grow them up in a common environment, they retain those color differences, which tells us that it's not all plastic change.
Yeah.
And then we also have some biochemistry data to try to understand like what are the compounds underlying these differences. So I would say yes, genetics and breeding within populations is very important for probably shaping these patterns. And if we look back, so we could look back at hydrophyllum, for example. So on this map, bright pink is hydrophyllum. Have this big cluster here in like around Milwaukee, and then we have a handful in like Northern Virginia. And the color differences in hydrophyllum, here are the data. So this is all the ones up
Yeah, you've got
Wisconsin, and these are in the east, and it's like, yeah, those are unlikely to be sharing a lot of genes.
yeah.
It's that gives us some evidence that yes, like genetic structuring probably contributes to this.
Okay. Well, I
And then
think out of the plants that you have, no, I do spend some time up in West Virginia in the mountains, but about the only one of those that I really see is the Houstonia cerula. So,
hmm,
I don't know how much help I could be with your project up there, but it'd be a little bit further west than some of these ones you have if you want that.
we would definitely anything. Yeah, so we have a lot of Houstonia cerulean, but I think like we were saying, there's those big geographic gaps. So
Yeah,
be happy
yes.
to send a card out and get more of that. Yeah.
There is another Houstonia there, but it's not the one you have, not Pusilla.
Yeah, we tried to pick really broadly distributed species that show this. We knew they showed variance between kind of whitish and purple.
Yeah,
Um, and so, yeah, I think in West Virginia,
yeah,
I,
the
I,
tiny blue, it's quite a
I
bit up there.
are hydrophyllum up there.
They are in a lot of places. It's just I spend a lot of time on the high mountains, and so I don't see much up there. But I there's places where I go once in a while where I might see it.
But, yeah, and then we added
And the lobelia might
that
I
might be
might see
there.
some of that too.
Fall bloomer, I think, late summer.
Yeah,
Yeah.
yeah. There's places where that's around, and it's around Winchester also. So
Yeah, we have very few of that one population, so would love to get more of that.
okay, yeah. If you want to send me a card, that'd be great. I could probably work on this a little bit.
Great.
Be interesting.
Um,
I'll go on your website and put in address and so
looks
on.
like take form, right?
Yeah, well, you can even text us through the Gmail, and we can ask you for the details. But there is an inform, like an inquiry, right? That you like uh, put your data that we ask, and we directly receive an email with all your data, your information, and we just
Okay.
email to you.
All right.
Great. Um, yeah, Richard, I don't know if you've been can you hear us still? Kind of popped in and out. Anyway, thanks for being here and listening to some of the preliminary results coming out of this. We're really excited to keep adding to it and there's going to be a lot of analyses that we still do. For every population, we're going to probably be extracting climatic data too, so we can link color with broad climatic patterns as well. So lots to go, and we'll keep you posted probably maybe once a year as we get new data in. So
Sounds good.
all right. Thank you.
Thank you so much.
All right. Thank you.
Bye.
Bye-bye.